Good morning. Well, I'm really excited about the topic today. This is a brand new lecture for me. When I learned about the topics for each of the weeks of the next-gen sequencing module, I was a little unsure what to do with myself on Amplicon sequencing week. So I found myself thinking, what do we do for bioinformatics in this space? And so I've spent a good week now just digging through what papers exist out there, what data resources exist out there, and I think I have some pretty good news to tell you about what's available in the algorithms to support Amplicon sequencing. So we're going to start with a talk about Pcr primers. Everyone loves designing Pcr primers, right? Mm-hmm. Well, there are tools out there to help you, and we're going to talk about a couple of them. We talked briefly about base calling error probabilities. You may have remembered my saying something about Fred when I was here last week. I thought it would be worthwhile to go just a little deeper into that because culling out sequences with sequencing errors is pretty important when it comes to doing Amplicon sequencing. So we'll cover that in a little more depth and the tool FastQC. From there, we'll talk about the great fun of operational taxonomic units, OTUs, and we'll have some discussion of diversity and how we actually generate OTU data starting with a pile of reads. From there, we will talk about the deleteriousness of non-synonymous single nucleotide variations. Isn't that fun? So having found a variant sequence, how do we evaluate its biological impact? And finally, we will talk about quantification and cases in which Amplicon sequencing has real challenges for us to deal with this class of data. Okay, so on we go into the fine world of Amplicon bioinformatics. So I thought I would start with kind of an old school paper. I'm 1993. This would be the year after I graduated high school, but I realize it might be a little earlier than that for some of you. But what is it that we use as our set of rules when we're trying to create a set of Pcr primers? So first off, you must employ primers that melt at similar temperatures. Does the reason for that make itself clear to everybody? Okay, so if you have a primer that's really GC rich versus one that's really AT rich, and you try to do PCR with both, you're going to find that the GC holds on a lot longer. It doesn't melt at as low a temperature as the AT rich primer does. So targeting the same level of the same temperature, the same degree of binding between the primer and the sequence is necessary. Do design target-specific primers. Maybe this is, again, obvious. You want to have a primer that doesn't just connect to any old place in your target DNA, but rather to one specific place. So that's very, very critical. Design efficient primers. Now, I've said near twofold, which is to say that each cycle you run your PCR, you ideally will double the number of your target sequences that you've got. If you have an inefficient primer, then your doubling will be reduced to maybe, say, 1.6-fold or 1.8-fold or something like that. And that can cut into the amount of expansion you get for the target sequence you're looking for. If you have a sequence that inhibits Taq polymerase, that's a problem. Obviously, we rely on that polymerase to generate our duplications, and so if we're inhibiting it with the sequence, that's bad. And don't allow for substantial homology among primer sequences. I'm pretty sure I mentioned the cocktail party problem last week. Does everyone remember what that's about? Cocktail party problem. How many different people in a cocktail party can shake hands? You can't shake your own hand. If A shakes B's hand, B has shaken A's hand. So that number is n times n minus 1 divided by 2. That's the total number of handshakes you can have in a party where n is the number of people attending. In the same way, if you have a very large number of primers, the number of handshakes that are possible among them, or between pairs of them, rises essentially with a square of the number of primers you've got. So the more targets you're trying to PCR at the same time in the same mix, the greater this problem of sequence homology becomes. All right. Now, I borrowed this paper, I borrowed a figure from this paper here, and we'll talk about what the the software product that describes it in just a moment. What they've done is to use these black and white arrows to represent primers with different targets. So the black pairing is a different one than the white pairing. So we don't want to have homodimers. We don't want a primer that interacts with itself. That's kind of like a worst case scenario. Nor do we want heterodimers where different primers are interacting with each other in pairs. We don't want end interactions where we have, maybe you don't have substantial homology within the sequence, but at the ends you do. That would be very bad, because you end up with interactions like that. You don't want the formation of hairpins like this. And you don't want to create situations like this where a primer from a different target interferes with the amplification of another one. So being able to deal with this class of problems gets very, very egregious. Now, if you were doing a really intensive Amplicon experiment, how many different targets might you incorporate in a single PCR amplification? I don't know, I'm asking this because I don't know. I don't do PCR all that typically, but I know a lot of people do. What's that?
What's the question?
Oh, okay. How many different targets might you go after in a single PCR reaction?
Yeah, what PCR are you using?
Would you go for 50? Not 50. What's that?
Yeah, if you multiplex it.
Yeah, I'm talking about multiplex PCR, yeah.
No, I don't do multiplex it, but yeah. No,
Okay.
you probably could.
Okay, so the more targets we go after, the more difficult challenges like this become. So this is principally a problem in that case. Now, oligo has gone through a whole lot of different versions. I've cited a paper from 1989. This is even older than the paper from 1993 we were looking at just a minute ago. So 1989 would be right after I started high school. I was learning to drive around the time this paper came out. Now, oligo has gone through a whole lot of different editions over time. It's now been through, version 7 is a very widely used one at this point. They are not just to be used for multiplex PCR. They can also support stuff like siRNA. Everyone knows what siRNA is? Really? Oh, that's interesting. Okay. It's silencing. So if you're trying to knock down the expression of a particular gene, you can create an RNA that is complementary to the messenger RNA for that, and that prevents it from getting translated. So silencing RNA. So if you want to do applications like that, or do restriction analysis or whatever, oligo is your good friend. So oligo.net is the software. This is, I believe, a commercially available software. This is something that we don't talk about a huge amount in bioinformatics, but it's something we think about, sure, certainly, which is to say that some software is commercially supported and closed source, and essentially, if you're going to be able to do something with that software, it's because the developers behind that software have given it that capability. But you, as a user of that software, don't have a lot of power, perhaps, in directing where that toolkit is going to go in the future. On the other hand, you're probably going to be able to get commercial support for it, so that's lovely. So on the left, we have that option. On the right, we have an open source toolkit for this, the Primer 3 software. So an open source tool doesn't come with support like that. Generally, their support is dependent on a community of users rather than some company with people paid to provide support. And if you really hate some feature of it and feel that you could do it better, you can actually make changes to it yourself. So from a bioinformatics perspective, there's actually a pretty big gulf between commercial and open source tools for the same purpose. Now, I would note that you may have used Primer 3 without even realizing it. If you've made use of the Primer Blast service over at NCBI, something is powering that website, and behind the scenes they have Primer 3 running. So this is, I think that this little dichotomy between these two toolkits has a lot to teach us about kind of the practice of bioinformatics in general. Commercial and open source tools may exist for the same purpose. Do we have a fair comparison between the two, an apples to apples comparison, which of these out-designs primers? Maybe not. but we do try to do that in lots of cases where we have competing tools. So as I move through these slides, you're going to see lots of cases where there are lots of tools doing the same thing, and we need to have some way to evaluate them. Okay, now let us talk a little bit about sequencing quality. So I mentioned Phred scores, and probably one of the first things you notice when I put this slide up is that it's deeply old school. Everyone, can you see the tiny little print up there at the upper left-hand corner, what this is coming from? It's coming from an ABI Prism Model 377. In the days of the Human Genome Project in the late 1990s, people would really have liked having this kind of instrument around. Today, it would kind of be stuck in an embarrassed corner of the sequencing lab. And so what I'm showing you is an electropherogram produced from a Sanger sequencer. So one of the old school capillary electrophoresis, fluorescence, and 4-channel stuff going on here. So when you see a black peak at a location, that's telling you that a G is likely to be found at that spot. If you see a green peak, it's an A, a blue, it's a C, a red, it's a T. All very straightforward, I think. So I would have you look at the ends of the sequence. What do you see all the way up here at in say less than 30 nucleotides into the sequence? Just a big blob of nonsense. I mean, you can't really get any usable sequence from that at all. Now, I want you to note that the resolution, the distance that these peaks appear from each other, gets a little worse as we start up at, say, 70, where you have baseline resolved bases, nucleotides. The peak for this nucleotide rises and falls all the way to the baseline, and then the next one rises and falls to the baseline again. When you get up here, look at, say, the stretch about 573. See this run of A's? Does it ever get down to the baseline again? No, you just have this. So you get these unresolved peaks corresponding to individual nucleotides. So it should be apparent to you that not every letter coming from the sequencing reaction is equally well known. So the people behind Phred, one of whom sat on my PhD committee, isn't that strange, were very invested in this problem. They wanted to have a way to estimate a letter-by-letter error rate. Now, from a political perspective, this actually had huge importance because when Congress in the United States funded the Human Genome Project for the United States parts of it, they argued that no letter of the sequence should be less, that there should be a level of accuracy associated with it. They didn't want just a bunch of sequence only to discover it was all irrelevant to us. They wanted to have some confidence that the individual base calls actually meant something. So they put in a requirement that the nucleotides be known, I believe, to an accuracy of greater than 1 in 10,000 errors. So that meant that we needed to have statistical models that allowed us to evaluate the quality of these base calls. So Phred was their answer to this. Now, just because I'm showing you an electropherogram from a Sanger sequencer does not mean that the way that we do this now with next-gen sequencers is any different. However, in typical sequencer operation, we do not collect electropherograms anymore with next-gen sequencing because these data are big. If you were to keep an electropherogram for each of 1 million different 100 base paired reads, you would have a lot more information to store than just the sequencing reads. Now, those electropherograms are generated on the fly, but that information is not stored to an output file like this. So I can show you what an electropherogram from a Sanger looks like, but generally that information is not available when we run a sequencing experiment over on the MiSeq. Okay? So the same rules apply in both cases. These data I'm just using to illustrate the point. Okay, so what is Phred all about? This paper was published in 1998. I had started grad school pretty recently and was discovering that seasonal affective disorder is for real. Living in Seattle in winter is harsh, guys. I'm just saying. No sunlight, makes me shudder to think of it. But great city to live in. All right, so how can we estimate individual base calling errors? First off, we need to infer a sequence from the electropherogram. We have to have something that says these letters go with these positions. So we want to associate each base call with a probability of error. Now, this is condensing 2 papers down to 3 very short bullet points, so I hope that this part will make sense. A peak must match the beat of its neighbors. So we have some regular spacing that we expect in these electropherograms. We've got a peak here, we've got a peak here, we've got a peak here, we've got a peak here. In bioinformatics, almost everyone who studies something associated with frequencies, with recognizing spacing in a signal, is using Fourier transform, sometimes called FT. Has anyone seen FT used for some purpose? Fourier transform? Well, let's say you've been to a friend's house party, and it's your friend the audiophile. Maybe he's playing house music or some kind of awful dance music at the party, right? So now, what's the most characteristic feature of dance music?
Dancing.
That's right, the beat. You're going to have something like a good whomp, 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 whomp going in the background. All right, so that is a regular beat. So if you were to look at this in a graphic equalizer, you would see this big spike showing up in that part of this frequency spectrum to reflect that you have a repeating signal in that space, at that frequency. In the same way, Fourier transform helps us to recognize what is the beat of an electropherogram. Whomp, 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 whomp. So that periodicity of the signal helps us to say, this is how far apart we expect these different base calls to fall in this electropherogram plot. All right. Next, only one trace is concave down at this call. Does everyone remember concave and convex? Yeah. Well, this is nice. Okay, so if you have a pebble, say, that's a convex surface. But if you were to if you were to think about a cave entrance, right, that is a concavity. It's concave space. So what we're asking then is, does the signal here form a U, which is concave up, or does it form an arc, like a rainbow shape? That's concave down. So we're going to ask about the concavity of the fluorescence signal here and expect it to be concave down, and we're going to require that only one of these traces is making a concave down hill, a hump at this space. Right? If you have A and G both producing a big spike there, that could be an error, or it could be that you have heterozygosity in the sample that you're working from, right? So this concave down property helps us to guard against multiple signals arguing that I'm a C or I'm a T all at the same time. Next, highly probable errors are far away. This is the argument that if you have a base call that's got a lot of doubt associated with it as a neighbor, you're probably also a problem. So when I lived in Nashville, I moved into a neighborhood called 12 South, and 12 South was an up-and-coming neighborhood, right? But I learned after the fact that the house that I'd moved into had been flipped by a real estate developer. They bought a trashy house and rebuilt a very nice house in its place, but before I moved into it, that neighborhood had been the crack house. So, right? So if your house is close to the crack house, you're going to have a lower property value than if you're miles away. And in the same way, if this base call within your sequencing read is very far away from the nearest error, likely error, you can feel more comfortable about it. So all of these factors are taken into account when we're trying to assess the probability of error at an individual site. I went over this a little bit last week, but I want to have a slide to talk about it too. We use a lot of negative logarithmic scoring in bioinformatics. And the reason why we do this is that proximity to zero matters a lot. When we're looking at probabilities of error, most of them are going to be in this space of much less than 0.1. So being terribly accurate about whether it's an error of 50% or 60% doesn't matter to us very much. But being able to say that this error is 1 in 100 or 1 in 1,000 matters to us tremendously. So that's why we use this negative log scoring, because it really explodes that space right close to zero. So if we have some probability of error that we call P, we can take the log base 10 of that and multiply that by negative 10. and that gives us the quality score for this particular position. So if you have a probability of error of 1%, that's the same as 1 times 10 to the minus second. Everyone remember that? The negative, remember that a negative exponent just means you're basically putting it all, putting 1 over that. So probability of 1% is 1 times 10 to the minus second. We give that a quality score of 20. A probability of error of 1 in 1,000 becomes a 30. A probability of 1 in 10,000 becomes 40. So these scores, as we mentioned last week, are really essential when it comes to doing our sequence assembly. They also matter rather tremendously in Amplicon sequencing. Okay. Now, FastQC is one of these really widely spread tools that we use for quality assessment in these data sets. And I wanted to show a plot from it to give us an example of that. FastQC is able to run through all the sequences in these FastQ files that we receive back, and from them evaluate how far you can go into the sequence before you start racking up huge error probabilities. In this case, we're working with Illumina 1.9 encoding. This is some Illumina sequencer, and it's producing short reads. The reads here are about 50 base pairs in length, when suddenly the probability of error has risen when we're looking across all of the reads that we have for the sample. So we want to keep that median error, the red bar across the yellow, really, really high. Having an error rate of 40 is all very tolerable. Remember that amounts to about 1 in 10,000 missed calls. By the time we get to position 72, we see that the probability of error has come to a quality score of 35, which is getting a little sketchy, right? It's not as high as 1 in 1,000, but it's still higher than we would like. And sometimes we have some very low quality scores indeed in this space past 50. So this is published 2013. There are papers earlier than that. Mostly I try to dig up the URL that is for the original tool. In this case, though, I'm getting a citation for a paper that evaluates the necessity of quality filtering in getting a stable diversity, phylogenetic diversity, from a sample using 16S rRNA. So this really starts to bring us into this world of dealing with microbial communities and trying to evaluate the extent to which a community is diverse or very narrow. Okay. I really dithered on whether to show this or not.
That's the latest, one of the latest memes.
It really is, it is, yes. I've seen a whole lot of different takes on it. And I don't want to put forward the stereotype that men are only staring at pretty bodies all the time. I don't want to forward the stereotype that girlfriends are shrewish. I mean, that's not true. You know, the people are people. So that said, I thought this was funny, and it was so on point that I had to include it. You understand? So, all right. So here we have the microbiome researcher. It's another white male. I'm sorry about that. But this microbiome researcher has been working with 16S primers for a very long time, and they've worked really well for a long time. And now there's a new technology about. Must the microbiome researcher turn away from this tried and true 16S technique and switch to metagenomics? All right. This is a real conundrum in the field of amplicon sequencing. Do we stay with this technology that we've used for such a long time, focusing on this region of high genetic diversity for all microbial genomes, or do we start sequencing more broadly to learn about the broader biology of the samples, not just which species are there? Because you're not going to learn a lot about the biology of individual organisms by simply noting that they're present at a certain level, right? So this distraction is something that we have to worry about. And I want to point out that if what you care about is which species are present in this sample, going after the more narrowly targeted 16S sequencing makes your data more interpretable than if you simply have a hugely diverse collection of proteins or of genes from all these different species. So don't be distracted. Focus on tools that you know work. Don't be caught up in thinking that your life will get better if you just do something that's newer. If you don't really know how to take advantage of the data from the newer technique, you may be safer going with the old. Okay, so there are a couple terms that show up aggressively in this literature, and I think that, well, for example, if I were writing a quiz question about today's lecture, asking what does OTU stand for would be kind of an obvious question, because OTUs are something that a lot of this field rotates around. So, what is an OTU? It is an operational taxonomic unit. It is a cluster of similar sequences, usually within 97% similarity of each other, represented by a single consensus sequence. And now I've put in really sloppy language in this at the very end of this, and I want you to be aware that this is how most people think of it, and it may not actually be that accurate. But we think of one OTU as representing one species. Sort of. Okay, so having sequenced a whole bunch of reads from the 16S region, we look for the most common sequences that we have here, and we ask if we can cluster similar sequences together. So we use this rather arbitrary criterion of 97% similarity to drag these together and say, well, I know that there's a little bit of sequence walkabout among these, but we're going to assert that these all come from one species, and the sequence variation that we see among them is just normal variation, or sequencing errors for that matter. Okay, so an OTU says we have this particular bug present. Now, diversity. You will very frequently see the terms alpha and beta diversity used in connection with microbiome samples, where we're trying to evaluate the sequence diversity that we have. So the most typical ones that I see mentioned are alpha and beta. And in this case, the word wrapping worked out just right. Alpha diversity represents the amount of diversity we have within an individual sample. And beta diversity tells us how much variation we have among different samples, different groups actually. So that's a gross simplification. If you were to read this 2010 article, I think you'll see that some of the math that we would use for evaluating diversity is actually quite a lot more complex than that. So we think of the diversity in a biome as being the gamma diversity, and that is subdivided in different regional into say different sub niches in that space. We might ask, we've got 5 samples from this area and 5 samples from this area. What's the diversity within each of those? What's the diversity from as we move from one niche to another? So that's where we would start talking about beta. So generally speaking, if you're trying to show that the microbiome upstream of a village is different than the microbiome downstream of the village, knowing that a lot of people do their laundry and do all sorts of other things in the water, you could look at the diversity differences between those two to show that some species are not observed upstream and are observed downstream, as an example. So that would be a look at the beta diversity between those two different sites. Okay, so these articles are useful in understanding those terms a little further. If you want to understand them better, I would suggest reading something like that. But how do we get to OTUs? So there are a fair number of tools out there for translating your sequencing reads into OTUs. And I'm going to talk about just these 3 for now, and we'll also look at the databases we use. So I want you to think of this as 2 parts. One is that we have the algorithms that are operating on the sequences that are doing the lookups and so on, and that's what we're looking at in this slide. In the next slide, we'll be talking about the reference sequences against which we will compare. So both of those are choosable aspects, but sometimes you see that people who always make use of Chime, for example, the second one here, always make use of a particular database for doing the lookups. So the 3 I've highlighted are all pretty significantly broadly used. This is not a complete list by any stretch, but these 3 are pretty popular. So we start with Mother. Mother was designed from tools called Daughter and Sons, which had been created back when everyone was doing Sanger sequencing. Now that everybody had shifted to using next-gen sequencing for this purpose instead, Mother was really needed to be able to scale to very large size. So even if you had 100,000 reads, you could still process it on a laptop, right? It's pretty important. So being able to scale well was something that they really hit at. So histogramming distinct sequences. One of the things that we operate from here is being able to count how many different sequences do I have for 16S rRNA. So I can enumerate all the different sequences that I see at this particular location, and I can count how frequently each of those has been observed. All right, so that's the histogram we're talking about. Maybe you have 10,000 different sequences, but you can account for 90% of your data with only 120, right? So something like that may happen. So that's this histogramming process. Next, we need to be able to cluster those at different distance thresholds. Now, just a moment ago, I was talking about this 97% similarity rule that's used kind of arbitrarily in this community. But what if you wanted to see how much diversity you had if you required the sequences to be 99% similar? I mean, if you compare any two humans in this room, you're not going to find below 99% similarity, right? So why would we expect microbes to be so much higher in their diversity? Well, there are very good reasons. Can someone give me a reason why? Why would you expect a microbial community to be more diverse than humans? Okay, well, it's a complex question. I would submit that bacteria divide a whole lot faster than we do, and as a result, they're able to explore a lot more evolutionary space than humans are. I mean, we're doing pretty well if we have generation spacing of, say, 20 years, right? But microbes are doing this every 30 minutes. So having greater diversity in a microbial population might be more likely. Anyway, 97% is the rule that we mostly use. Mother is designed to evaluate what this clustering would look like with all kinds of different boundaries, though. So that's a helpful feature. And it does a fair bit of reporting on the diversity that comes out of it. I also have to say, they have a sweet logo. Take a look at that thing on the upper left-hand corner. Doesn't it look like someone caring for the data? I think that's just really beautiful. All right, well, anyway, so they have a good graphic artist. They have pretty good software. They published early 2009, and they own a fair bit of the next-gen sequencing space. So Chime is a little different. Chime reminds me, in some respects, of the OpenMS project in proteomics, in that they've really tried to create a plug-and-play system where if you want to try a different system for clustering or try a different similarity assessment system, you can make those you can change those components to do so. So it's a modular pipeline of these tools designed to work in one seamless workflow, and they've done a fair bit of work on visualization and metrics. Now, how do we feel about their graphic artist, though? See that logo at the upper right? That's Chime. First off, how many people knew before today that it's pronounced Chime? Right. QI does not always strike everybody as being a chai sound, but there you go. So that's Chime. Their tool set is available right there. I've given the citations right down here at the bottom. So the 2009 paper is Mother, 2010 is Chime, 2013 is UParse. That brings us to UParse, which is also part of USearch. Now this is a curious little guy. When I was looking at the article associated with this, it was, first off, surprising to see a single author paper. I mean, when was the last time you wrote a paper that had just one author on it, right? It's not that common an occurrence. But this fellow, R.C. Edgar, is in business by himself. He runs his own software company. He produces tools explicitly for next-gen sequencing, and UParse is his baby. That's his gig. Now, he has, I borrowed this figure from one of his manuals, I think, but it's not in fact a logo for his company. I haven't seen that one. But he had a very clear notion of what he wanted to accomplish. First off, he argued that a sequence that you see only once is not a sequence you care about. A sequence you see only once is not one you care about. So if you have a single sequencing read, he says, this is noise, throw it out. If you have multiple copies of a sequence, then you can start to trust it. That's his view. So his histogram, we talked about that up on Mother, remember? We had how many times each sequence had been seen. His solution is chop out any singletons, then start work. So that's the first part of uparse. The next is the problem of chimeras. Chimeras. What is a chimera and why are we concerned about it? Okay, a chimera is a sequence that spans two different species. In doing your sequencing, you might have had some funky business with maybe some odd ligation events and any number of random bits that happened when we do molecular biology. You may have had a case in which a sequence from one species was pasted onto the sequence of another species, and then it got amplified. This is a chimera. So do you see why that's problematic in a clustering algorithm? A chimera that combines a segment from A and a segment from B is likely to look similar to the A and to look similar to the B. And suddenly your clustering goes haywire because these chimeras are dragging together different bunches of sequence. Now these other toolkits also deal with chimeras, but they tend to do it after clustering to look for these mistakes after the fact. But uparse is designed to do that in the course of clustering. So all three of these tools are out there. Some of them are free, some of them are not. Great. So if we have the ability to do this clustering and to do this noise suppression that we've talked about by getting rid of chimeras, getting rid of single reads, whatever you want to do, we still need to be able to say, what does this cluster of sequences represent? I love living in a country that drinks tea. Such a good thing. All right. So there are 3 databases that I've selected here. Are these the only databases out there for looking up 16S RNA sequences? Not even close, not even close. But these 3 are pretty big. So RDP, I have to say their art is pretty good. I mean, that's pretty tight, right? Nice and compact. It scales well. You can have it at a very small size and still read it. That's nice, right? Green genes. Oddly enough, LBL did not. Lawrence Berkeley Livermore Laboratories did not make this logo show up when I hit their website. Maybe they're just having a down day or something. But this logo is out there, and it's a nice one. It's the 16S rRNA gene database and tools. Always nice when you can say you are the database, right? And then Silva, high-quality ribosomal RNA databases. Now, as we look at the top one of these, the Ribosomal Database Project, back in 1997, again, I just started grad school, has now, in the most recent release, I checked it yesterday, has 3.3 million 16S rRNAs and 126,000 28S rRNAs. What am I talking about 28S all of a sudden for? First off, what's the S stand for? Anyone know? The sedimentation, did I hear? Or did I hear the name? Svedberg. Svedberg was the person who studied sedimentation coefficients, and so the unit is named in Svedberg's honor. So, all right, so a 28S is larger, so it moves through the gel more slowly. So we've got we have 126,000 of those. We study 28S rRNAs when we're interested in fungi, right? We think of 16S rRNA as being our way to go after microbes in general, but there are plenty of microbes out there that are not bacteria. So that gets us into the fine world of fungi. And so having the availability of rRNA from fungi, or for that matter from eukaryotes like mammals, etc., is something we can also do. The Ribosomal Database Project includes both. It should make you aware, in looking at those 2 numbers though, that our coverage in the world of bacteria is a lot larger than what our current coverage is in fungi. Now, does that mean, is that only because there's a greater diversity of bacteria in the world than there are of fungi? Or is it because we don't study fungi as much as we do bacteria? We could be missing part of the biological story. So the coverage that you see in these different branches of taxonomy may reflect what kind of information we can get out of the databases when we do this mapping process. Okay, so ribosomal, the RDP is nice and large. Green genes, however, is also quite large. they have also been concerned about this problem of chimeric spectra. Imagine the problem that, we already talked about why it's bad to try to do clustering when you have chimeric sequences present. Imagine if your database also has chimeric sequences present. That's a big problem. Now you may mislabel a species A as species B just because a chimera for both is present in your reference database. So doing a good job at curating these databases is necessary to protect us when we go out there using green genes from misclaiming that one species is actually another. Okay, and Silva is also very popular, 2007. It grew from a toolkit called ARB. In an earlier version of the prior slide, I'd listed ARB as one of them, because ARB also does this clustering and lookup business. So you can imagine ARB as a fourth item listed on the prior slide. But Silva grew out of that, and they have large and small subunit databases available to work with. All three of the papers appear down here. There are also papers out there that evaluate the coverage of these databases against each other. I used some of those to figure out which ones I wanted to highlight here. So before you do your OTU assessment of sequencing reads, it probably will behoove you to read a few of those papers to ask, is the database I'm planning to use to look up my sequences well covered in the species I care about? Okay, so on we go. Oh, we get to do some statistics today, just a little bit. All right. Rarefaction curves are very frequently used in the space of phylogeny estimations. So I've sampled some stream water, and I've taken a look now at what 16S rRNAs I can find in that collection. I've looked them up. But let's say I get back a list of 536 OTUs. Something I've totally omitted so far is the degree of specificity we get out of OTUs. How often is an OTU that we get from green genes all the way down to species level? Has anyone looked at data sets of this type? Very, very frequently, you're doing very well just to get genus, let alone the species. So a lot of the time, the OTUs that are produced by a lookup in green genes or RDP or whatever will not be all the way to the species level. So being able to work with results from these tools can also be problematic. You know, I've tried to find what is an appropriate proteome database for searching a proteomic sample if I know the 16S derived OTUs. But if I don't know which species is there and only know what genus is there, things get a lot harder to work with. Okay, now, rarefaction is a way to evaluate whether the list of species, the list of OTUs that came back is complete, right? Maybe I needed to do twice as many reads to get a proper assessment of that. So some of the first papers that we saw in the microbiome didn't really look at ordinary communities. Instead, they looked at things like acid mine drainage, because acid mine drainage is a very harsh biochemical climate in which microbes can live. And so you needed to have, so the acid mine drainage environment would have relatively few species for you to look at. If your sequencing depth were able to pick up every species that's present in a mixture, that would be brilliant. But we always feel that we are sampling that environment. We're not getting an exhaustive assessment of those species. So rarefaction curves come to the rescue. In effect, rarefaction is making your data worse to see how much the findings degrade. Everyone got that? We are going to subsample from our sequencing reads, use just a fraction of them, in order to see how stable our OTU findings were from the whole. So we can ask, how many species do we have if we start with our original data, which is represented by the right arm of the right side of each of these curves, and how much it shrinks as we cut down the number of sequences that we produced, that we allow into the OTU assessment. So we can see that there's always some amount of curve. So ideally, you would like to see that the curve has gone asymptotic, that we have a a pretty stable number of species that even if we doubled the sequencing, maybe the number of species would rise by 10%, but not much more than that. That would be great. If you see that the value is just streaking upwards as you get to the full set of data you produced, you can probably have produced twice as many sequences and produced a much, much larger phylogeny as a result. So rarefaction curves are for that purpose. So to rarefy is to take a random subset of a given size of the original sample. In statistics, you would probably see that called bootstrapping. Have you seen that term?
Yeah.
Bootstrapping? Okay, so rarefaction is a kind of way to do that. So rarefaction curves can be used to understand the depth of sampling of a community compared with its total diversity. Very challenging articles in this space that suggest that we're never going to be done sequencing everything that's out there. I think that's already pretty clear. But as you read other papers in the rarefaction curve space, one of the things that you conclude is that statistically it's kind of problematic, that you always want to use all the data that you have, and rarefaction, of course, cuts into the data we have available to us. Okay, now, having found a sequence variant, we would like to know whether that's a meaningful one or not. So I'm going to step away from 16S for a moment. I've spent a lot of time yammering on about that. Instead, let us imagine that we are sequencing, genes associated with drug resistance in infectious disease, something like that. So in a case like that, I have some known gene from this species. Normally, the wild type sequence for this location has an S present. What I've observed, however, is a T at that position. Does it or doesn't it affect the function of that gene? Right. So, first off, what's this term I'm using in the header here? The NSSNV. Well, I'm glad I stopped then. Okay. So, this is a non-synonymous single nucleotide variation, or variant in this case. So, generally speaking, if a mutation in a genetic sequence creates a silent mutation, we don't care very much about it. Generally, that's weak, but it's true. We tend to focus our attention on those mutations in genetic sequence that change amino acids, or that do things like induce a stop codon, something like that. So non-synonymous means changing the resulting amino acid sequence. SNV means it's a mutation at an individual position, or it's a sequence variant at a particular position. Everyone good with NSSNV? It's not an easily pronounced thing, but it's very helpful when you're trying to write a paper on the subject. Okay, so is it deleterious? What does deleterious mean? Not necessarily a deletion. Does it invoke a deletion? But is it harmful? Is really the context in which we use that term here. So is a mutation harmful to the function of this particular protein? So there are three algorithms that I highlight here. Again, there's a world of them out there, but these three are pretty widespread. Now, I mentioned the BLOSSOM database when I was here a week ago. Everyone remember BLOSSOM? It's a substitution matrix to say, how painful is it if I replace a serine for a threonine? How likely is that in the scope of evolution? So Hennekoff is back. Ung and Hennekoff back in 2001 published their paper on SIFT that used this concept of sequence homology to ask how frequently replacing this letter with another letter would be in the context of evolution. So given a particular sequence and claiming a variation at a particular spot in that sequence, SIFT will then grab a whole bunch of sequences aligned to that query sequence and ask, was it or wasn't it dangerous to modify this residue? If it's a very highly conserved site, if it's always S in functional proteins in the wild, we start to think, well, changing that for a T is probably going to mess with it, maybe perhaps by steric hindrance or something, by being very slightly bigger. Okay, so SIFT does this almost entirely around the concept of sequence alignment. In the wild, does the sequence letter change or not? Polyphen, coming out in 2002, really takes a more, it adds this element of structure to it as well. So if there are, if this query sequence is homologous to a sequence for which we have a known structure, we can ask whether this mutation is taking place in a highly ordered region or a region where sort of anything goes. You know, if you're sitting out in a loop that doesn't really interact with anything, changing an amino acid is not going to be terrible. If, however, you're in a transmembrane domain and you change a leucine into a proline, you know, you've just abolished the hydrophobicity at that position and put a big kink in the chain. That's not going to be very good for your transmembrane domain. So this 3D structural element is very useful. It is also able to use feature tables. So when you look up in UniProt, a particular protein entry, you'll see that some regions are helices and some regions are sheets, and this is active site, and this is a known site for phosphorylation, etc. That feature table is also useful. So even in cases where we don't have a homologous structure, we probably have something that says these are known domains of the sequence, and making changes within those can come at a greater cost. So SNAP was 5 years later, and it really reflects a lot of the changes that came about in computer science over that time, because machine learning, if you haven't been following bioinformatics as long as I have, you may not realize that there was a time when we didn't put machine learning on everything, right? Now it seems that you can hardly have bioinformatics involved in something without machine learning showing up somewhere. So machine learning was used to give it an ability to calibrate whether a change was problematic or not, and solvent accessibility got involved as well. Do people know what solvent accessibility is? Okay. When we attempt to decide how a protein folds up from its sequence alone, one of the features that we would use for that purpose is solvent accessibility, which is to say that the hydrophobic parts are next to hydrophobic parts. They're not on the surface of the protein where they would interact with the solvent in which it sits. So one of the ways that we evaluate whether a potential structure for a protein is good or bad is to ask how much of its surface area is now solvent accessible that wants to be, and that it has, say, basic or acidic residues, or that doesn't want to be, that's got leucine or valine and all these hydrophobic residues. So all 3 of these can be used pretty much freely. You can read review articles that ask how one compares versus the other. It's useful information. Up at the top, I've included an example, I think from Polyfen, if I remember. And so here we have just our little thermometer. If we have a score all the way on the right, then we think that this sequence change blows apart the function of this protein. It's just completely inactive. Over at the left, we say, no one cares. We changed a leucine for a valine. They're both hydrophobic. We don't care too much. So that's kind of the sort of information that we get back from them. Do they agree with each other? Do you think that they all produce comparable results? Just curious. There are people who would assert that, well, they're all designed for the same purpose, so their results have got to agree pretty well. But in fact, most graduate students who've run 2 or more of those systems on their data sets discover that they can have rather different claims about the same sequence changes. So it's valuable to get a second opinion in this case, to understand whether those results are robust across the algorithms. How am I doing on time?
1 hour in.
1 hour in, okay. Hey, we're doing pretty well. We're going to finish up maybe 1:15. That's my guess.
That's nice.
75 minutes, okay? Okay. I can tell people are kind of getting draggy over there. If you need to shake it out, do it. It's all right. I do it all the time up here. I'm moving around. It's hard to do that. All right. I wanted to give you 3 examples where Amplicon sequencing was valuable for quantitative exercises. Okay? So I'm going to start with tuberculotic sputum, right? Somebody has developed tuberculosis, and we want to know what variant of critter they've got growing in their lungs. So we're going to have them hawk up a pretty nasty one, and from that, we're going to ask what fraction of the tuberculosis bugs in their sputum are resistant to TB drugs, right? So maybe we have about 10 genes that we think are most common sites for the induction of tuberculosis drug resistance. Right, there's a ribosomal protein, I happen to know that one, but I don't know the others. That's fine. So we would like to know what fraction of the critters growing in this person's lungs have likely tuberculosis drug resistance. So we're now going to create a primer set that allows us to multiplex out 10 genes known to be associated with this drug resistance. And we're going to ask, do they or do they not have mutations thought to render this protein no longer a point of attack for our drug? So, did you see that first part of the phrase there? What fraction? This is a quantitative question. So we need to know whether the number of what basically from the starting sample, what fraction of those critters had these mutations. Because there's a diversity of microbes growing in that person at this time. We need to know what fraction of them are dangerous. It might be that we just give this person conventional treatment for the drug, and that we see that we do a great job of killing off all of the bugs in their lungs that have the ordinary sequences. But we see that with every month we give them this normal treatment, the fraction of resistant bugs grows, right? Important clinical question, and it relates strongly with what we've been talking about through this entire lecture. Next. Now we're talking clinical biology. Now we're talking about a tumor in cancer. So I'm tracking this tumor. I have biopsies from this, say, skin tumor over time. I want to know, do we see key genes that are known to be associated with what we call driver mutations in cancer? Are those genes accumulating driver mutations over the scope of months as we watch this person in their progression with cancer? Right? Finally, do species change in dominance within this bacterial community as a function of pH? So maybe I have a stream before it reaches the factory and a stream after it reaches the factory, and I want to know, do I see, maybe I see the same species both before and after the factory, but I want to know, are those, are we changing in dominance by dumping lye into the water or dumping acid into the water? So again, all three of these are quantitative questions that we want to address. This area seems to be a fair bit, a bit of a Wild West right now. So this is going to be a lot more associated with general principles of quantitation, but I hope that it's still useful. So you've heard the term sigmoid curve, perhaps? Sigmoid? Yes. All right, this is an example of a sigmoid curve. So we have some amount of DNA really in our sample, and we have a detection that we're using read count for this example. And we want to know, does the number of DNA read counts rise as the amount of DNA rises? Yes. Yes. In this case, we have a rise in read count as DNA concentration rises. However, if I have 0 reads, it does not mean that DNA sequence is absent in the target sequence. That is because of the level of detection. We have a limit of detection, essentially, that says below this concentration we shan't be able to observe it. Now PCR is really powerful, but in most of the analytical technologies we use, we have some lower boundary that says if you have 1 copy of a thing or less, you're not going to be able to see it, right? So generally speaking, we do not assume that just because we have 0 reads that there's no presence of the thing. Next. And this is an important one, I think, the level of quantitation, that read count does not rise linearly with too few or too many sequence copies. There is a strong temptation for people in working with quantitative technologies to always assume that this slope, this spot in the middle, is where your data must fall. You shouldn't assume that, though, because if you have too few copies, you're not getting an increase in you're not getting an increase in what your detector says from these very small counts. And up at the top, you have this kind of blatting, right? Do you ever turn your stereo all the way to the right? Turn the volume knob all the way to the highest level? You don't get as much of a gain between, say, 9 and 10 as you do between 6 and 7. Part of that is because your ear is doing some work of screening out really loud noises. So detectors, whether they're mass spectrometers or sequencers or whatever, tend to have this point of saturation where adding more signal does not increase the readout that comes from it. So anytime you're trying to estimate these, don't assume that just because you have 5 times the read counts, you have 5 times the DNA in the target DNA in the sample. We have to be careful about knowing where this range of linearity is, where our read counts are in fact useful for inferring concentration. So why else might read counts be a terrible way to work? When I say read counts, by the way, I mean that very literally. We have DNA reads from our sequencer, and we're just counting how many times we have a hit on this particular subsequence. So it could be that we've done a multiplex primer assay, and some of those primer efficiencies were close to 2, and some were closer to, say, 1.5. That's a bad scenario, because now those sequences that were associated with highly efficient primers have been amplified at a much greater rate than those that had less efficient primers. So you can't do an apples-to-apples comparison in that case and say that we had this number of reads for this sequence and that many reads for the other sequence, therefore we conclude that sequence A was present at a greater concentration in our samples. Because our amplification was biased towards one rather than the other. Sequencing errors may cause us to believe sequence variants exist that do not. So there's always going to be a problem with sequence quality in next-gen sequencers. If we have a particular variant sequence present, it might be that it was biologically present, or it could be that the sequencer is playing a little game with us. Okay, stochastic noise. Do people feel comfortable with the word stochastic? Stochastic. What I used to teach my biologist friends is kind of rude, actually. I teach them that if you want to say random without sounding like your technique is arbitrary, you should call it stochastic instead. Stochastic is a stochastic process is one example is the light switch, right? Every time you flip on the light bulb, you have some chance of blowing the filament out. Old school light bulbs, Edison light bulbs, right? So if you can't predict exactly when a particular light bulb will snap, but you can say in the mean, I expect these light bulbs to last through 10,000 switch flips, for example. So stochastic noise is stuff like that. It may cause us to believe cohorts are different when they are not. And the experiment that I always end up proposing to people is a highly conservative one. Mix together some reference sample, a whole bunch of it, and then run it through the same sequencing technology multiple times. Sample the same, sequence the same sample 5 times, for example, and ask, how much variation do I see among the reads that I get from these different experiments with exactly the same sample? Analytical technologies are almost always somewhat variable among technical repeats like that. And misunderstanding that technical noise is actually biological signal is a very bad thing for us. So if we are trying to assert that this particular subpopulation of microbe is growing by 20% from point A on the stream to point B on the stream, if your technical noise is 30%, your claim is built on rubbish, right? So stochastic noise may cause us to believe cohorts are different when they are not. That shows up all over the place. All right, lots of takeaway messages, but I was really pleasantly surprised to see just how much bioinformatic thought has gone into supporting Amplicon sequencing. We start with selecting Pcr primers, some very long-lived tools there in that space that have seen a lot of tinkering over time to improve their functionality. Controlling the quality of sequencing is always necessary when we're dealing with NGS. So using a tool like FastQC, I didn't mention it, but one called Trimomatic that uses these quality scores to figure out how to lop off the ends of reads is available too. Trimming the error-prone flanking sequences, we mentioned that. Clustering reads to determine the distinct set of sequences. This is kind of the first step towards getting us a list of OTUs. Annotating sequence clusters with taxonomy information. In my experience of having looked at microbiome data sets, that's almost always the weak leg. We have a lot of sequences that don't get fully resolved species names. We may have just a genus, or we may have just a class in some cases for these. So getting solid taxonomic annotation on our clusters is almost always a point of weakness for these experiments. Determining the completeness of sequencing, how many species might be present in this sample that we didn't see because we didn't sequence enough. Right, that's an important one. That we talked about that with the rarefaction curves. Evaluating the disruption of function by mutants by non-synonymous single nucleotide variations. Maybe I should ask what that abbreviation stands for. That might be straightforward. All right. Quantifying particular sequence variants. I wish I had more to say that we had statistical models that were highly robust against every type of error in that space, but I cannot say that yet. So I'm really glad to have gotten this chance to learn a little bit about Amplicon sequencing myself. Next week, we will be talking about RNA-seq. Oh, we're going to talk about some statistics next week. It's going to be a lot of fun. So everyone needs to know a little bit about statistics. But I think that's the end for today. And I'm happy to take some questions if any of those have shown up in flight. All right. Well, if that's the case, I'm all done with you for the day, and I'm glad to have worked with you. Thank you very much.
Thank you so much.